So I first introduce our first speaker, Colonel Dr. Mithu Banerjee. Ma'am is additional professor and head in biochemistry, AIMS Jodhpur. She has pursued her MBBS and MD from Armed Force Medical College, Pune. Ma'am has worked in Army Medical Corps for 24 years and has been nominated as chair of Diabetic Testing and Harmonization, Asia Pacific Federation of Clinical Biochemistry. Ma'am is also representative from India as corresponding member in IFCC. Dr. Banerjee has also pursued a postdoctoral fellowship at prestigious Johns Hopkins Medical Institute in cancer epigenetics. She was instrumental in starting work in cancer epigenetics in armed forces. Ma'am was nominated as Fellow of Royal Society of Biology, and Ma'am has numerous publications in national and international journals. So I now invite Dr. Banerjee for her talk.
Yeah, thank you, Anisha, for those kind words. So, warm tidings from the blue city of Jodhpur, and that's the institute and aerial view of the All India Institute of Medical Sciences at Jodhpur. So, yeah. My topic today is, are your genes your destiny? So most of us are inclined to think that we have a set of genetic code, and our genetic code decides phenotypic outcomes in our lives. But epigenetics is the science which has actually kind of disproved this theory that you are endowed with a set of genes and your genes determine the outcome always. So therefore, the name of my talk is, are your genes your destiny? The world first got sensitized to epigenetics when in Times there was this article by Mr. John Cloud, who was a public health specialist who was working at the Karolinska Institute, and he called his article, Why Your DNA Isn't Your Destiny. So this, I will be covering the lecture in these subheads. Going back to the story which was published in the Times magazine. So the main work was done by one Dr. Lo Bygren, who was working at the Karolinska Institute. And he went on to describe a story in which Norrbotten was a village in Sweden. And here the population density was very, very sparse. So and it was very geographically isolated. So when there was very bad harvest, people would starve, and on certain years when there was very good harvest, there were people who would overeat and engage in gluttony. So he made an Dr. Lo Bygren observed that those villagers in Norrbotten in Sweden who had indulged in overeating, when they were followed up, their kids and grandkids died 32 years earlier than those who did not indulge in overeating. So therefore, from this conclusion, when he followed up these families over years together and decades together, he thought that there was genetic imprinting of overeating which was left in the sperm and ova, and therefore these traits of metabolic risk and metabolic disease had passed on to their progeny, and therefore they died 32 years earlier compared to those whose grandfathers had not indulged in overeating. Another experiment which was conducted by Randy Jertle, he was an oncologist, and Robert Watlang in the Duke University. What they did, they had two cohorts of mouse with the agouti gene. Now this agouti gene gives the phenotype of a yellow coat to the mice, and it predisposes them to metabolic diseases like diabetes and obesity. Now, they took two groups of agouti mice. They were pregnant, both cohorts were pregnant. To one cohort, they fed vitamin B12 and folic acid rich diet. And to the other one, they just gave a normal diet which was not enriched with vitamin B12 and folic acid. Now, we all know that vitamin B12 and folic acid are methyl donors. So what happened in the cohort in which vitamin B12 and folic acid was given, there the mice which were born, they did not have brown coats, they did not have yellow coats, and they were not obese. In other words, there was something in vitamin B12 and folic acid which underexpressed the agouti gene. Therefore, the mice which were born, they were having a brown coat and they were having normal weight. Unlike when agouti gene was being expressed, their coat was yellow. Here what had happened, their yellow coat had changed to brown coat and they were of normal weight. They did not suffer from diabetes, nor did they suffer from obesity. This is the picture. This is the normal weight, normal mice, and this is the when agouti gene was expressed. This is how they looked. So the father of epigenetics is Dr. Conrad Waddington, and he's a very person with a multifaceted interests. He was an anthropologist, a paleontologist, a biologist, and he is a person who first coined the term epigenetics. Now, epi, as we all understand, is over and above. So epigenetics would basically mean the science which dictates the phenotypic outcome without a change in the genetic code. We all know that when the genetic code changes, there is a change in the phenotype. But here we are talking of the genetic code remaining same, yet the phenotypic outcome being different. And how does that happen? That happens because of this epigenetic changes. So to define, there are changes in gene expression that are heritable, that is, they are passed on to they can be passed on to the next generation, and they are heritable, and they do not involve change in the DNA sequence. The DNA sequence remains constant. So if we were to give an analogy, genome is the hardware and the epigenome is the software. So the epigenome eventually determines which gene should be expressed at what point in time, which gene should be repressed at what point in time. And these phenomena occur in normal embryonal development also. So we have this phenomenon, like if we were to take identical twins who are from the same zygote, during infancy their environments are more or less quite similar. But over the time they move out, their environments are different and they might have different phenotypes. We know about twins, identical twins who in whom one of them has schizophrenia, one of them doesn't have schizophrenia. So how is that explained? That is explained due to the environmental factors or the epigenetic changes which they have, although they're developing from the same zygote and hence they have the same genetic code. So therefore, this basically drives home the hypothesis that nurture drives nature. So when we want to start looking at epigenetics, we just do a quick recap. This is the chromosome, as you can see. And the chromosome is tightly wound around these protein discs, which are known as histone discs. And the genetic material is all encoded in these DNA. And you can see over here, there are from these histone discs, there are certain histone tails which are jutting out. Now, some factors, some chemicals, they get attached to these histone tails, or they could get attached to these DNA tails. And when such flagging or tagging of these molecules occurs, this leads to a different kind of outcome, phenotypic outcome. So what all could happen if there is a genetic change, epigenetic change? It could lead to cancer, it could lead to a host of autoimmune diseases, mental disorders, right from schizophrenia to autism to metabolic disorders like diabetes. And so when we read about epigenetics, it could mostly occur at two levels. One, that DNA could be modified. The DNA that we saw was round around the histone, that could be modified, and we know that there are 4 nucleotides. Most commonly, cytosine is methylated. That means to the cytosine there will be a methyl tag which is attached. So the DNA could be modified. So this becomes an epigenetic change. The code does not change, yet there is a little flag on the code which either represses the gene or expresses the gene. Similarly, the histone discs that we saw, they could have a lot of tagging. They could have an acetyl or methyl or phosphoryl. So similar, lot of chemical modifications could occur on the histone disc itself. So this is how the histone bodies are basically of 4 types, H2A, B, 3, 4. And you can see that these alphabets over here, P, E, P, V, K, all this, these are all coding amino acids. And when I was saying there's a tagging here, you can see K is a lysine and there's an acetyl molecule which has got tagged on the lysine. So therefore this has got modified. Similarly here you will see that on this lysine there's an acetyl and a methyl. So there's a lot of modification which is occurring. Here you will see again, this lysine has got acetyl and methyl tagged to it. So this is how chemical tagging occurs in the nucleotides. Although there is no change in this nucleotide sequence, the fact that they could be tagged differently by chemicals could change their expression. So one was about the histones and the DNA. Now, very recently we have come across miRNAs. These are basically nothing but these are that part of the genetic code which is not coding, right? And they are responsible for post-transcriptional regulators of gene expression. That is, after the mRNA is formed, it tags those mRNA and hence produces different kind of molecules which in turn produce different kind of outcomes. So these miRNAs act by, what are these miRNAs? Functionally, what do they do? So these are small sequence of nucleotides. These mRNAs act by binding to the main messenger RNA. From the DNA, we first get a transcribed messenger RNA. This messenger RNA is acted upon by miRNA. It binds to the mRNA and it causes the destruction by small molecules, which could be an siRNA or an RISC. These are all small molecules with the aid of which miRNA destroys the miRNA will destroy the mRNA. Hence, there will not be any translation and therefore there will be no protein product formation. Now, there could be methylation of the messenger RNAs as well. And this mostly happens in the adenosine nucleotide. That is, at the 6th position, there could be methylation. And similarly, the cytidine could also be methylated. Cytidine is mostly methylated at the 5th position. RNAs also could be methylated and they have their own enzymes which are methyl transferases. That is, it transfers a methyl group and they have their own set of demethylases. That is something which removes a methyl group. And now mRNAs are being explored massively and they've been, they are the very potential source of biomarkers and they also have a lot of therapeutic potential. So how is it that this chemical tagging of the histones or the DNA, it can help in repression or expression of a gene? So if we again were to look at these DNA which is tightly wrapped around the histones, you see over here that there is just one methyl when it is euchromatin. Euchromatin means it's nice, widely spaced out, right? But when there is hypermethylation over here, you see there are at least 5 methyl tags on the histones. When this has got hypermethylated, it all compacts. It becomes very compact. The chromatin becomes very, very compact. And therefore, the transcription factors cannot come and fit. And there is no expression of the gene. So basically, with hypermethylation, the euchromatin becomes heterochromatin. It's very tightly packed, not letting the transcription occur because transcription factors will not be able to come and sit. Now, looking at the machinery of how methylation and demethylation occurs in the body. So here again, we are back to those discs which are known as the histones, and here you have the DNA. If you look over here, this there are certain writers, that is, they transfer a methyl molecule. So this K is nothing but the code for lysine. So we could have a lysine methyl transferase. What does it do? It transfers a methyl molecule. This blue is a methyl molecule. So it has, this is a writer, that is, it is methylating. An eraser. What is an eraser? Again, so this one is a KDM. That means it's a lysine demethylase. So it will remove the methyl and it will make it unmethylated, right? Similarly, when you look at this histones, acetylation, we talked about acetylation. Now here if you see, HAT is histone acetyl transferase. So what has it done? It has removed and it is adding an acetyl group to the tail which is jutting out from the histone. So it's a histone acetyl transferase. And then the erasers over here could be histone deacetylases. So what has it done? It has just removed one acetyl and it has deacetylated. And then you have these enzymes which are erasers again. They remove the methyl group, TET enzymes, which have again a lot of potential. They are being explored massively. They are nothing but the 10-11 translocases which remove methyl groups. So eventually, the methylation would be a tight balance, a tightrope walk between the writers and the erasers. When the methyl transferase levels are high, there will be methylation which will be occurring. When the erasers or the TET, the 10-11 translocases are high, there will be hypomethylation. So eventually, this is a game of balance between methyl transferases, demethylases, acetyl transferases, and deacetyl transferases. There's lots more to it, but to make it simple, I'm confining myself to this. So it's basically like when you do not have epigenetic tagging, this is just a set of nucleotides which are there. But when there is tagging with, say, methyl groups or acetyl group, the cell exactly knows which gene should get expressed and which gene should get repressed. And then this string of notes gets converted into a beautiful symphony. So that is the difference. This chemical tagging actually determines what should get expressed and what should get repressed. Now coming to the specific of cancer. In cancer, it has been seen that we have this pictorial of this gene over here, and near the promoter region, there is a very rich concentration of cytosine and guanine. So these regions are known as CPG islands, right? When in the CPG islands, what happens? The C is contiguous with the G. So when the CPG island, the CG rich areas of the promoter region of a gene are not methylated, the gene expresses, right? This is unmethylated. Now, when the CPG island has got hypermethylated, what happens? There is repression of the gene. The gene does not get expressed, and you can well imagine that if this were to be a tumor suppressor gene, say like P53 or retinoblastoma gene, if these were to get hypermethylated and repressed, it would mean that the process of carcinogenesis would go on unabated because the tumor suppressor gene has now gone down-regulated. Now, the beauty of this whole thing is that this whole thing is reversible. Like I said, it's a tightrope walk between methyl transferases and demethylases. So here you see this is a cytosine molecule, and this cytosine molecule basically with the help of a methyl transferase, DNA methyl transferase, it gets methylated at the 5th position. At this 5th position, there will be a cytosine. And there are certain drugs or molecules like azacitidine, which the subsequent speakers will delve on in more detail. These molecules inhibit the DNA methyl transferase, and therefore methylation cannot occur. And this whole thing is reversible. The 10-11 translocases, they reverse this methylation which is occurring. Now, in the lab, how will we study methylation? So the main strategy which we undertake is bisulfite modification, and there are n number of techniques. I will just run through them. When you want to look at specific genes, that is, you want to study gene-specific methylation, you could do a conventional PCR, you could do a real-time PCR, you could do a restriction fragment length polymorphism. Now with high throughput techniques, there is phylosequencing, which is very convenient for small sequence. You could do it, enriching with chromatin immunoprecipitation, and there could be NGS, which is done. So the whole thing revolves around doing a good bisulfite modification of the extracted DNA, and then depending upon the objective of what you want to study, you could choose a technique. So what exactly happens in bisulfite modification? How does it help us differentiate between a methylated and a non-methylated cytosine? So here you see this cytosine molecule. This does not have a methyl group, whereas this one has a methyl group over here at the 5th position. So when we subject DNA to bisulfite modification, there's a whole lot of steps which you need to do to modify. There is predominantly deamination. That means this amino group is removed and it is replaced by a keto group. So this cytosine actually becomes uracil. But however, when this 5th position C has a methyl group, it is resistant to bisulfite modification. So this is a very beautiful strategy to differentiate between methylated cytosine which resists bisulfite modification, and unmethylated cytosine which undergoes deamination, and thereby the cytosine becomes uracil. So coming to gene specific methods, we all know for a conventional PCR we will have to do a DNA extraction. Then we showed you bisulfite modification. It's some 10-15 steps. Then we have primers specific for a methylated, specific for unmethylated, and a wild type. And eventually we make a gel, run the DNA and see. This will only be a qualitative detection of methylation. So this is one of the gels that we had done. In real-time PCR, if it's probe-based, it's much more specific. So we can only do with M primer. We do not need to have a U primer here. And this will give you a semi-quantitative methylation status. So here, if you can see on these curves, this is 100% methylation when we see the CT value somewhere near 23. Here there is 50% when the CT value is around 25. And with zero methylation, the CT value is around 28. So that is how real-time PCR would look like. What about pyrosequencing? Now, for pyrosequencing, for all these methods, we have to first do a bisulfite modification. So again, we have done here bisulfite modification, and this reads by detection of a release of a pyrophosphate when a particular nucleotide goes and joins. So we put ATCG. So if there was an AT would go and join and a pyrophosphate would be released, and that could be detected. It's quite similar to Sanger sequencing, wherein a dideoxynucleotide incorporates and the chain stops termination. So basically what we would do, we would first run a DNA which is not bisulfite modified, and then we would run the bisulfite modified DNA and then compare and see whether the C has got converted to P or it remains C. If the C remains C, that means it was methylated. If the C has become T, that means it was not methylated. So this is how this would look. Other methods of enrichment of methylated DNA, this is chip immunoprecipitation, wherein this is the methylated DNA. It has been sonicated to cut into small fragments which are manageable. Then we denature it. That means we make them single-stranded. Then we expose this to methylated antibodies. So there is tagging of this methylated one gets the antibody attaches to it. And so we have a very enriched fraction of methylated DNA because we've removed the unmethylated ones. And then we could do a microarray. As you can see here, this is a microarray, or you could basically run an NGS or a Sanger. So this is one of the enrichment methods, a chip immunoprecipitation. Or again, there could be a RFLP, restriction fragment length polymorphism, wherein we use certain enzymes to cut. So there could be a methylation sensitive restriction enzyme. So what is this? That means if there is methylation, it will not cut. So here you see the red is methylated and the black is not methylated. So since it is methylation sensitive, wherever there is methylation, it is not cutting, right? It could be a methylation insensitive restriction enzyme. Basically, that means that it would go and cut anywhere. It is insensitive to methylation. So it has cut everywhere. Or it could be a methylation dependent restriction enzyme. That means it will cut only when there is methylation. So you see wherever there is methylation, it has cut. And then after it cut, we look at the fragments on agarose gel electrophoresis, and this would give us whether there is methylation or no methylation. Now, that being said about the techniques, the epigenetic research boom is in the US. It was said that in 2011 it was worth $800 million. In 2013 it rose to $1,000, and in 2017 it became almost 10 times of what it was in 7 years, from 11 to 17, $8,000 million. Unfortunately, I could not get this figure for 21 or 22. So I'm showing you old figures of 2017. So after having done this, I would like to just present our data of one set of prostate carcinoma study that we had done. And here we had taken 84 cases of prostate cancer, 30 normal controls, and we had 7 who had high-grade prostatic intraepithelial neoplasia. So basically what happens, this is cancer, then we have HGPIN, and then we have control. So this is in that sequence. We took serum from all of these patients, and we took 16 primary matched tumor samples from these cases. A quantitative methylation specific PCR, that is a real-time probe-based chemistry, was used, and we did the methylation specific PCR to obtain relative quantification of these 10 genes. So when we look at, since we had done it from serum, we wanted to be sure that our assay was okay. So we first looked at the, we wanted to check out the analytical sensitivity of QMSP from serum. So here you can see the enzymes that we had done. First, we looked at how many tumor samples. We had taken 16 tumor samples. So we wanted to see how many of these genes showed methylation. So over here, you can see that out of the 16 tissue, 13 showed methylation for SSBP2. So on, if you see MCAM, all 16 tumor samples showed methylation. Similarly, if you look at RAR beta 2, all 16 samples showed. So that is 100%. Now, when we want to compare what happened in the tumor with the serum, we looked at methylation from serum divided by methylation in tumor. So over here, if you see ER beta, when we performed QMSP from the serum, two samples showed methylation out of these 13 tumor. So we did concomitant serum from these 13 who had shown methylation from the tumor. So this is the kind of sensitivity which we saw we had got when we did QMSP from serum. So you can see over here, out of 16 samples which had shown on tumor, none of the serum samples showed methylation sensitivity. Over here at CCND2, out of 16 samples, we see only 2 had shown. So serum was showing methylation pattern. So looking at this, this is the serum versus tumor. So all these are the 10 genes, and we've done from serum and tumor. So here you can see that there is over here, mostly you'll have methylation in the tumor and much less in the serum. So this is a pictorial depiction. Now, looking at the sensitivity and specificity, when we took MCAM, ER alpha, ER beta, and SSB2, what do we see? We see that there is a sensitivity of 78% and specificity of 53%. When we removed SSB2 from here, the sensitivity was 75 and the specificity increased to 70. We had done serum PSA for all of them, and as you can see, the PSA has a sensitivity of 77%, but specificity was poor, only 30%. So therefore, this proved that if you were to do MCAM, ER alpha, and ER beta methylation, you would get a good specificity of 70%, whereas what we get from prostate is just 30%. Sorry for the interruption, my throat is... So this basically what happens, if we could do methylation of these 3 genes, we would unnecessarily obviate a lot of prostatic biopsies. Here what happens if we only do PSA, it's got a specificity of 30%, and we all know that even with a rectal examination, the PSA level goes up. So therefore, there was an advantage in doing methylation of these 3 genes. Looking at the levels, if you see look at MCAM level, this is the level of methylation, then comes HGPI and slightly less, and then controls. And if we were to look at early stage tumors, because that is where you would want to detect, late stage anyways, there is no problem in detection. And if you look over here at early stage, this was the methylation level, and in controls, it was much, much less. So clearly, MCAM is helping us differentiate between prostate cancer and normals.
Excuse me, ma'am. We are exceeding time.
Yeah, I'll just take a minute. I'm just finishing.
Thanks, ma'am.
There are some FDA-approved diagnostic tests which have come based on DNA methylation. So one of them is the MGMT methylation, which is in glioblastoma multiforme, and that when this is present, temozolomide is an effective way of treating. GSTP1 methylation has been used for cancer, so is APC methylation. And interestingly, when vimentin methylation is looked at from stool in colon cancer, it can help with the prognostication of advanced CRC in terms of hepatic and peritoneal metastasis. So these are genes which help in MLH1 methylation helps in colon cancer, and SEP19 methylation in colon cancer has been correlated with reduced overall survival and disease-free survival. So finally, to conclude, the beauty of detecting DNA methylation is that it's a reversible thing, and therefore it offers a very good opportunity for therapeutics. And hypermethylation of promoter genes is mostly seen in cancers, and demethylating agents and deacetylase inhibitors will play a very major role in precision medicine in the days to come. But there are many hurdles from when we want to take them from the bench to the bedside, that the next speakers are going to allude to that. Thank you.
Thank you, Dr. Banerjee, for your illuminating talk.